If you hung on, if you are back. Now we're going to look at proteins. Now, again, this is the activity that we're going to do in class this week. We're actually going to look at, because they're a little more involved, we're going to look at the structures of proteins, and we're going to build some models of them in our short little class this week. Excuse me. But before we do, we want to look at the protein is kind of the coolest one because it's the most structurally and functionally diverse. As far as functions, this list could probably go on for quite a ways. But I kept it kind of with what the book had said and sort of what you have maybe talked about or have heard about before. So if we look at this list, this first line, things like pepsin and DNA polymerase, there's a blast from the past, I hope, right? One of those enzymes involved in DNA replication, right? So one of the functions, enzymes, or proteins are enzymes. Keratin and collagen, if you've been watching TV and seen lotion ads or anything like that, that is what those are talking about. It's part of the structure of skin. So proteins are involved in structure. Hemoglobin, aquaporin. Again, hemoglobin, you probably recognize that from discussions of blood. Aquaporin, maybe. That is the thing within your cell membrane that allows water to transport freely through the cell membrane. It's a special carrier that transports just water, so that's aquaporin. So they are involved as carriers and transport. All right, and then we have a large category, which is going to be cell communication, which is one of the new units this year we're going to take a look at for you guys. How do cells communicate? So there's things like insulin and other just general hormones. Those are signals. Then we have the other end of that, which is the receptors, which then receive those signals and do something with them. In terms of defense, immunity, right? Big topic of the day right now. We produce proteins that are called antibodies. For movement, your muscle fibers have what's called actin and myosin. They basically work like these little Velcro pieces that grab onto each other and do this and release and do this, and that's how you flex a muscle. And then something for like a plant, like a bean seed protein, proteins can actually be used as storage. So they're super, super diverse. There's a lot of stuff going on. We're going to look at a few examples, but before we do that, we got to look at just the general structure. So when you look at a general structure, we did this with the other ones. In this case, we have monomers and polymers. The monomer of a protein, in other words, the building block, are called amino acids. All right, and you guys may remember this. There are a certain number of them, and you'll never have to remember them, or memorize them, but there are essentially 20 different amino acids that make up the human proteins that we have. So the monomer would be the amino acid. The polymer would be a poly for many peptide. Right? And then when you do that, you take proteins, you fold them up, and they get really big, they get really complex. Rubisco, this one right here, is commonly studied. That has 16 different polypeptide chains lumped into one big one. Hemoglobin has 4, 1, 2, 3, 4, 2 alphas and 2 betas. The growth hormone, so there's all kinds. The biggest one, there's that silly video a lot of you saw. I don't know, Mr. Strick showed it, but it's a guy reading the name of the longest word in the English language, and it's a protein shortened up to called Titan. But he like grows a beard while he's reading it. It's kind of a goofy one they saw on YouTube. In terms of amino acids, it has different parts, right? So if you look at what I've got here in black, we would have what's called the central carbon. Right, and that's that central portion which has just a single H on it. That's the central carbon that everything else is built off of. Excuse me. In blue, we have what's called an amine group, or amino group. It depends on who you talk to, but that's the portion in blue. In green, we have what's called a carboxyl, and again, I know we built these with our functional group thing the other day. And then we have what's called an R group. And that's a kind of a side chain. And there could be 20 different side chains, right? So you can see our little penguin here. He gets it. Oh, NH2, that's the amino. And that is carboxylic acid. So amino acid. makes the name, right? So when you look at these, the structure, and this is what the lab will clarify for us here, is it has different levels of structure. So one of the things that's important is what we call disulfide bridging, right? And it's a unique part that makes up and helps us to form different structures of larger proteins. So they form these things called disulfide, so two sulfide bridges, right? So if you look at it, when the R group has, right here, when the R group has a sulfur in it and a sulfur in it, they actually will form a bond, right? And sulfur, if you ever remember what sulfur smells like, rotten eggs. So if you know anyone who's ever gotten perms, like probably not a big thing in this society of today, but when I was in high school, people get perms and their hair would smell for a little bit, kind of like rotten eggs. If you went by a beauty shop that was doing them, it'd smell like rotten eggs because they're breaking and building these bonds that have sulfur in them. So that's going to stabilize the 3D structure. So that R group comes into play, I guess is my point. The R group is going to make them do different things. So you might have a disulfide bridge, but in reality, what we're looking at is if you look at a single amino acid, here's one, and then here's another one. Those two are going to take water out to make a bond that forms right here. It's a very special bond. It is called a peptide bond. So it's a special type of covalent bond that is between the NH2 or the amine of one amino acid and the COOH, which is the carboxyl of another, right? And it is a C to N bond. It's right there. C to N bond is what it would be, right? That is called a peptide bond. And of course, the process, we already know, is dehydration synthesis. All right, so we got that. So they grow, and they always will attach from the what they call the N group of one to the C group of the other, right? The COOH group to the N group of the other. So they grow in one direction from N terminal to C terminal, right? That's how they're growing. So when you look, okay, polypeptide chain, this is what I was just saying. Polypeptide chains have one direction. The N terminus is the NH2 end, and the C terminus is the COOH end. So it's a repeating sequence of N, C, C, and then it goes again. In other words, if you look down here, N, C, C, another one, N, C, C, another one, right? This would be the N terminus, this would be the C terminus, so it grows in that direction. People always ask, why the Star Trek? If you ever look at Star Trek on the Enterprise, it's the NCC Enterprise. I don't know if that's why, but I don't know. Maybe science nerds, maybe that's what they did. So in terms of structure, what is it? Well, it is the function, and this is something that we always say in biology, function depends on structure, or function, or structure determines a function. So in this case, what we do is we take it and we twist it and we fold it and we add it to other ones that are twisted and folded, and that gives us our large piece, okay? If you look, we basically have these four parts, and this is what we're gonna do in class, right? We have our primary, we should change that to black. So we have our primary, we have our secondary, we have our tertiary, and we have our quaternary. So 1, 2, 3, 4. The primary structure is a sequence of amino acids, and that is determined by the DNA. In other words, the DNA codes for what order they should go in, and the primary structure basically looks like a beaded necklace where the beads are different amino acids, and that's it. Then, when you get to the next step, when you go from primary to secondary, we now take that beaded necklace and we cause some hydrogen bonding. So if you look, it's either going to be a beta pleated sheet, so it's going to fold like that, those H's are then going to cause that pleating to stay, or it's going to make this alpha helix, and then the hydrogen bonds are going to cause it to hold that structure. So primary is just a single sequence. Secondary, now the hydrogen bonds hold it into those formats. Now the tertiary, they are going to be interactions between the R groups, and that is the key with these. So what you're going to get is you're going to get some hydrophobic interactions. And again, we're going to play around with this in class to clarify, because this gets pretty detailed. Interactions. So hydrophobic means water hating. So those are going to tuck to the inside of these folded up tertiary models. We may get some disulfide bridges. So if there's sulfur and sulfur, they're going to go towards each other and create a little bond. We're also going to get some hydrogen ionic bonding. So all that's going to happen in here to sort of lock it into that tertiary structure. And then the quaternary structure is just multiple tertiaries that are stuck together. And again, depending on the one it is, it could have 4 if it's hemoglobin, or it could have a lot more. It just depends. So again, primary, single chain. Secondary, either folded or helix because of hydrogen bonds. Tertiary, we now pull in the R groups, and they start to cause interactions to happen, either hydrophobic interactions, disulfide bridges, hydrogen bonds, ionic bonds, all of those things, and then put a bunch of those together, and we get the multiple tertiaries. And that's the polypeptide, big polypeptide. Now, I know it's a lot here, guys, but hit pause anytime you want, and then come back when you're ready to watch a few more minutes. In this case, proteins are going to do what's called denaturing. They're going to unfold, right? And that can cause problems. So we have these conditions that are going to disrupt those hydrogen bonds, the ionic bonds, and the disulfide bridges, and it causes problems for that organism. One of them is temperature. So if you are running a very high fever, the high temperature in your body takes that protein out of its optimal range, and it can actually disrupt the shape of that protein, therefore causing massive problems, right? That's one way that fevers can go bad. Changing the pH, making it too acidic or too basic, can actually cause the full protein to unfold. Changing the salinity, the body chemistry, can alter that as well. Right? If you alter the primary or the secondary and tertiary structures, you're actually altering the 3D shape. If you change the shape, you destroy all functionality of that thing. So think of it like a puzzle piece, right? If you destroy the shape of that puzzle piece, it can't do its job anymore. Now, some proteins, once they've gone back to a normal scenario, can return to their normal shape, but most cannot. Once you've deformed them, it doesn't work anymore. The example I'd always use in class, if this is my pen, I throw it in the microwave and I melt it, well, clearly it's going to deform into some other shape. It won't work anymore, but I can't reverse that. I can't unmicrowave the pen. It just doesn't work. So that's how proteins work. This I just pulled from your text. This is actually an example straight from there that shows those interactions. So this would be the stuff that happens on the tertiary level. Right? Here you have, that would be a simple hydrogen bond. So that's causing some folding to occur and stay. Over here, these ones are hydrophobic, so they don't like water. Hydrophobic interactions, so they don't like water. So basically, since the cell is the majority water, they fold to the inside of the protein to avoid that water. So those are called hydrophobic interactions. You'll also see something called van der Waals interactions right there. Here you see two sulfurs. So that one would be your disulfide bridge. And then over here, we see here, there's a slight positive and a slight negative. So those would be an ionic bond. So all of those are interactions at that tertiary level to cause that folding to occur. Now, once you have that, you then move it to the quaternary structure. So you take multiple, you fold them together, right? Collagen is that way. It's folded together. It's just another level. Hemoglobin, it's a very common one. There's 4 subunits. And again, just another example. So when you look at all of those, the 4th level structure results when 2 or more polypeptide chains form one macromolecule. So that's how you get the big quaternary ones, right? Collagen was one, hemoglobin is another, has 2 alpha plus 2 beta. They're just examples of ones that you're going to see. If you ever are interested, sickle cell disease that's what it is. It's a change in the primary structure of one. It's a DNA. It's a change so subtle, but it causes a massive change in the ability of the hemoglobin to be the correct shape, and it results from a single. This is crazy to me. A single amino acid substitution. In a protein hemoglobin. So single, that's crazy to me. It's just a single little thing. This is straight out of your text. It's just showing you that structure of hemoglobin. I just think this is really cool to understand it to that level. Here, the 6th one, it changes from GLU to valine instead. That causes this region to change shape. It exposes it. That's the hydrophobic region. It exposes it. Instead of being normal shaped, it has these sickle cell shaped. When it gets into its fibrous form, it causes that sickle cell rather than a normal shape. And it's crazy. It causes massive problems for people that have it, especially when they have it at a more affected level. The other thing about proteins, in addition to the primary structure, physical and chemical conditions can affect the structure as well. Alterations in pH, in salinity, we kind of already said these, temperature, they can all affect it. Loss of a protein's native structure is called denaturing or denaturation. And a denatured protein then is biologically inactive, right? It will not work anymore. Now, the last thing I will show you, and I'm almost done. There are times when they can go back and forth, but typically once it's been denatured, it can't renature, and that's a very rare thing to have it actually go back. But the one thing that cells do have the ability to do is they have to fold that protein correctly. It doesn't just happen. So there's one last thing, and you may remember this. There's a protein that actually works on the proteins to give them the correct shape. It is called a chaperonin. So I always think of the word chaperone, right? But a chaperonin is a protein molecule that assists in the proper folding of other ones. And the reason I bring this up, again, I'm trying to make this so you can see real life versions of diseases such as Alzheimer's is one, Parkinson's is another, where they have found what they think, mad cow disease, but they have issues with misfolded proteins, and that might be due to the chaperonins not working correctly. So basically, if you don't fold them right, then you have some effect that's going to happen. So the chaperonins need to do their job correctly. Last tiny bit I'll show you. Last slide. This is a chaperonin. This is from your text. Basically, I always think of it like a little shaker bottle, right? There's the shaker, there's the cap. You put the polypeptide down into it, so you stick it inside, you put the cap on, and you shake it up. Once you've shaken it up, it allows it. So basically what it does is the internal environment of this creates an environment so that it can fold correctly, and then as it opens up, it comes out correctly folded. So it's probably enough for you today. Again, there's a lot in there. Look at that chapter. It's actually a pretty interesting chapter if you're into like the biochemical, the bio macromolecules, nutrition, any of that stuff. This stuff is pretty fascinating. So take a look, take some notes, and we'll call it good. Thanks, guys.